In this presentation, we are going to summarize complete oxidation of glucose, starting with glucose all the way to carbon dioxide and water, and we will also look at some related topics. In the course of glycolysis, two molecules of NADH are produced. Aerobic oxidation of this NADH by the electron transport chain is preferable to formation of lactate. However, the inner mitochondrial membrane is impermeable to NADH. Animal cells have developed several shuttles to transfer electrons from cytoplasmic NADH to the mitochondrial electron transport chain. Two important examples of such shuttles are the Glycerol phosphate shuttle and Malate-aspartate shuttle. First, we will examine the Glycerol phosphate shuttle. Dihydroxyacetone phosphate, which is produced, as you probably recall, in the course of glycolysis by action of aldolase, is reduced in glycerol phosphate shuttle to form glycerol 3-phosphate. The reaction is followed by the oxidation of glycerol 3-phosphate by mitochondrial glycerol 3-phosphate dehydrogenase, which utilizes FAD as an electron acceptor. The reaction occurs on the outer face of the mitochondrial inner membrane, and glycerol 3-phosphate does not enter the matrix. FADH2, that is produced as a result of reaction, is then oxidized by the electron transport chain. FADH2 produces only 1.5 molecules of ATP per molecule of cytoplasmic NADH. The second shuttle that we are going to examine is malate-aspartate shuttle. It is a more complex and more energy efficient shuttle. The shuttle begins with the reduction of cytoplasmic oxaloacetate to malate by NADH. Malate is transported into the mitochondrial matrix and is then reoxidized by NAD to oxaloacetate. In the course of reaction, NADH is produced and it is then oxidized by the electron transport chain. For the shuttle to continue, oxaloacetate must be returned to the cytoplasm. As oxaloacetate cannot pass through the inner membrane, it is converted to aspartate in a transamination reaction with glutamate. So the glutamate aspartate transport protein transports aspartate in exchange for glutamate. In cytoplasm, aspartate can be converted back to oxaloacetate. The malate alpha-ketoglutarate transport protein transports alpha-ketoglutarate in exchange for malate. And then in cytoplasm, alpha-ketoglutarate can be converted to the glutamate. Glutamate aspartate transporter requires moving a proton into the matrix, and that reduces net synthesis of ATP. So instead of generating 2.5 molecules of ATP for each NADH molecule, the overall yield is approximately 2.25 molecules of ATP. ATP that is generated in the course of the citric acid cycle has to be transported into the cytoplasm. 2 molecules of ATP are produced in the course of the citric acid cycle from GTP. Their transport into the cytoplasm requires uptake of 2 protons into the matrix. This reduces total amount of ATP that is produced by about half a molecule per cycle. The overall process, depending on the shuttle that is used, produces between 29.5 and 31 molecules of ATP. Average is 30 molecules of ATP, which we can compare with only 2 molecules that are produced in the course of glycolysis. The net overall reaction for the complete oxidation of glucose is 1 molecule of glucose reacts with 6 molecules of oxygen and 30 molecules of ADP and 30 molecules of phosphate. Products are 6 molecules of carbon dioxide, 6 molecules of water, and 30 molecules of ATP. While electron transport chain is usually used for energy production, it could be also used to generate heat as needed. Under normal conditions, electron transport and ATP synthesis are tightly coupled and heat production is kept to a minimum. But newborn babies, hibernating animals, and cold-adapted animals require more heat than is normally generated by metabolism. In a specialized form of adipose tissue called brown fat, most of the energy produced by the mitochondrial electron transport chain is dissipated as heat. This tissue has brown appearance because of a large number of mitochondria in it. About 10% of the protein in the mitochondrial inner membrane of brown fat cells is unique 33 kilodalton protein called uncoupling protein, UCP, or thermogenin. When it is active, it dissipates the proton gradient by translocating protons. As proton gradient decreases, the energy is dissipated as heat. The process is regulated by norepinephrine. Norepinephrine is a neurotransmitter, which initiates a cascade mechanism that ends up hydrolyzing fat molecules or triglycerides. Uncoupling protein is activated by binding to the released fatty acids. Fatty acid oxidation continues until the norepinephrine signal is terminated or the cell's fatty acid reserves are depleted.